morning, students. Today I'm back with a brand new chapter, Animal Kingdom or Classification in Animal Kingdom and its basis. So this chapter is divided into two parts. Today we are going to learn part 1 and tomorrow or day after tomorrow we will be studying the next part of the chapter. So quickly let's start. Okay. So the first thing that we are going to learn about Animal Kingdom is basis of classification. Basis of classification. So what are the few primary bases of classification? Arrangement of cells, body symmetry, nature of coelom, pattern of digestive, circulatory or reproductive systems, and also level of organizations. Level of organization is actually a part of arrangement of cells. So now we will discuss all these bases of classification in details. So first point is level of organization. All members here are multicellular. So what we should remember is that in animal kingdom all the members are multicellular, but still they do not exhibit, remember this, they do not exhibit same pattern of organization of cells. Same pattern of organization of cells. What do they mean? See, a multicellular organism might not be arranged into a tissue. What is tissue? Similar type of cells will be arranged for a particular function, and that is known as tissue level of organization. But all multicellular animals might not be having a tissue level organization. Like in the previous chapter we were learning, no, ground tissue, epidermal tissue, those vascular bundles formed of xylem and phloem. So each are tissues formed of different type of specialized cells which are assigned some kind of specialized functions. So here what happens, all of the members are multicellular, but they might not be exhibiting same pattern of organization of cells. What are the examples? In sponges, what are sponges? Porifera. We will come to these things later. The cells are arranged as loose cell aggregates. The cells are arranged as loose cell aggregates, and they exhibit cellular level of organization. They exhibit cellular level of organization where division of labor exists. Like few cells perform few functions, few cells perform other, but this division of labor occur among the cells. It is not that well divided that the cells of a particular type performing a particular function can be divided into a discrete tissue. So the differentiation is not that advanced. Division of labor is there, but at the cellular level. Okay. Now comes coelenterids. In case of coelenterids, the cells performing same function or similar function are arranged into tissues. Hence is called tissue level of organization. See, I was telling you, no, in few organisms, the level of organization might be cellular. In other organisms, the level of organization might be at the tissue level. Okay. So till now we understood the level of organization. So till now we have learned till cellular level and tissue level. Now we are going to learn organ level. What is organ level? It is present in the members of Platyhelminthes and other higher phyla. It starts from Platyhelminthes and goes to other higher phyla. Here what happens, a particular tissue or a particular type of tissues are grouped together to form organs. Here particular tissues are grouped together to form organs. So first we found cellular level where we found in Porifera or sponges. Then in Coelenterate we found tissue level. Now in Platyhelminthes and other higher phyla we are finding organ level of organization. Now in these like annelids, arthropods, mollusks, echinoderms till chordates, here organs are also associated to form a functional system, and these are known as organ systems. These are known as organ systems. Okay. So, what are organ systems? We know very well that we have digestive system, circulatory system. So, these are parts of organ system. See, in example here also it is given, circulatory system. Circulatory system again can be of two types, like advanced level and the primitive level. In open type, what happens? In which blood is pumped out of the heart and the cells and tissues directly bathed in it. I mean directly bathed in the blood. Open type is in which blood is pumped out of the heart and the cells and the tissues are directly bathed in it. Like blood is pumped out of the heart and directly the cells and other tissues are as if drenched in that blood. There is no specific veins and everything, like veins will carry the blood to different organs. No, they are all drenched in that blood, and there will be diffusion or absorption of several things like oxygen and giving out of carbon dioxide, like that. So this is an open type. And what is closed type? In which blood is circulated through a series of vessels called arteries, veins and capillaries. So that is what I told. Closed type is where the cells are not directly drenched in blood. In fact, there the arteries, veins and all these things carry the blood to different organs, and then again that deoxygenated blood is carried back to heart, where it is again recycled or renewed. So this is a closed type of circulatory system. Now it is clear? Now another basis of classification. So till now we were studying which basis of classification? Level of organization, cellular, tissue level, organ level and organ system. Now we are coming to symmetry. Organ can be asymmetrical, like in sponges. Yes, like in sponges. Like this. See, where there is symmetry, there is no, if you cut like this, these two sides might be different. If you cut like this, these two sides are different. So it is asymmetrical. That is, any plane that passes through the center does not divide them into equal halves. Any plane, see, neither this plane nor this plane can cut it to equal halves. So this is asymmetrical body. Now we are seeing radial symmetrical body. Sorry students, there was a power cut off here. Okay, so now we are going to study about radial symmetrical body. This is what? This is when any plane passing through the central axis of the body divides the organism into two identical halves. Like, see here, this is a kind of radial symmetry. Here there is 1, 2, 3. Here there are 1, 2, 3. And through any plane, if it is cutting the body into equal halves. Is it clear? This plane, this plane, this plane. Let me draw a cross section of this. Like just the top view. Just the top view, okay? Just this view. See, this is and this is equal. See here, this and this equal. If radially we cut, so if we cut radially, then also it's equal. So this is a kind of radial symmetry, and its example is coelenterets, ctenophores, echinoderms. Its example is coelenterets, ctenophores, and echinoderm. Now we will be coming to bilateral symmetry. Bilateral symmetry is what? When the body can be divided into identical left and right halves in only one plane. Like this. See, if you cut it like this, then it is not giving symmetrical body. Only when it is cut through the center, it is giving two symmetrical halves. So, classification can be based on the type of symmetry. Now we will be coming to diploblastic or triploblastic organization. What is diploblastic and triploblastic organization? It is the number of layers we are having. Now this is diploblastic. This is diploblastic. This is triploblastic. What is more in triploblastic than in diploblastic is this mesoderm. This mesoderm, this red colored layer, this is the mesoderm. This is extra in the triploblastic, which is not present in diploblastic. Instead of mesoderm, what is present in diploblastic? Mesoglia. Instead of mesoderm, what is present in diploblastic? Mesoglia, which is not that much differentiated as mesoderm. This is an undifferentiated layer. This undifferentiated layer, mesoglia, is present between ectoderm and endoderm. Clear? This one. This is the undifferentiated layer. This red here in the diploblastic. This layer is present in diploblastic. Okay, and in triploblastic we know that here there is the embryo has a third germinal layer, mesoderm, in between ectoderm and endoderm. This is present in platyhelminthes to chordates. So now let us go to the next part, that is presence of coelom. What is coelom? Coelom is body cavity lined by mesoderm. Coelom is body cavity lined by mesoderm. So what we can understand? That only triploblastic organisms can have a true coelom. because mesoderm is present only in triploblastic. But all of the triploblastics also doesn't have a proper coelom. Sometimes they have pseudocoelom. Sometimes they have pseudocoelom. Like coelomates are animals possessing true coelom, like annelids, mollusks, arthropods, echinoderms, hemichordates, and chordates. And what are pseudocoelomates? These are animals where body cavity is not lined by mesoderm. Instead, what happens? Mesoderm is present as scattered pouches in between ectoderm and endoderm, like this one. See, like here. Let me change the color. Like this one. See, here there is a continuous mesoderm. Here there is a continuous mesoderm lining the body cavity. This is the body cavity. And this red one is the mesoderm. But see here. The mesoderm is present scattered here and there between ectoderm and endoderm. So these are known as pseudocoelomates. These are known as pseudocoelomates. And what is a coelomate? The animal in which body cavity is absent, like platyhelminthes. The animals in which body cavity is totally absent, like that of platyhelminthes. Here there will be ectoderm, mesoderm and endoderm. Clear? Ectoderm, mesoderm and endoderm. So this is the presence of coelom, pseudocoelom and no coelom. This is another basis of classification of animal kingdom. What is the another basis? It is segmentation. It is the segmentation, whether the body is segmented or not. Segmentation can be external and internal, with a serial repetition of at least some organs. With serial repetition of at least some organs. For example, earthworm. The body shows this pattern called metameric segmentation, and this phenomenon is called as metamerism. What is this earthworm body? It has segments, right? It has segments, and this phenomena is known as metamerism. Now, another basis of classification is presence of notochord. Presence of notochord. It is a mesodermally derived rod-like structure. Again, where it can be present? It can be present in triploblastic animals because it is a mesodermally derived rod-like structure formed on the dorsal side during embryonic development. So it is a mesodermally derived rod-like structure formed on the dorsal side during embryonic development in some animals. Animals with notochord are called chordates. Animals with notochord are called chordates, and animals without notochord are called non-chordates. So what are we? We are chordates. Okay. So this is another basis of classification. Now we will come to our first phylum, that is phylum Porifera. Now we are going to come to our first phylum, that is phylum Porifera, which is also known as sponges. These are the three, let me cross the preview. These are the three sponges here, Cycon, Euspongia, Spongilla. See here, can you see any symmetry? No. That is what we mentioned earlier, that in sponges there is no symmetry, hence it is asymmetrical body. So they are, see, it's written, mostly asymmetrical animals. They are mostly asymmetrical animals. They are primitive multicellular animals and having cellular level of organization. This also I explained what is cellular, tissue, organ and organ system level of organization. So these are primitive multicellular cell and having cellular level of organization. They have a water transport or canal system. They have a water transport or canal system. Water enters through minute pores or ostia in the body wall into the central cavity. This is the central cavity called spongocoel, from where it goes out through the osculum. From where it goes out through the osculum and it comes out through ostia. Comes in through ostia, goes out through osculum, and this is the spongocoel. This is the spongocoel. So they enter through ostia, goes out through osculum, and they enter the body cavity, that is spongocoel. Now, this pathway of water transport is helpful in food gathering, respiratory exchange and removal of. This type of water transport is helpful in food gathering because food comes through the water. Food sometimes comes through the water. Respiratory exchange of gases and removal of waste. Schwannocytes or collar cells line the spongocoels and the canals. Schwannocytes line the spongocoels. Digestion is intracellular. Digestion is intracellular. The body here is supported by skeleton made up of spicules or spongin fibers. See, here we have a primitive level of skeletal system. Although a primitive level, but still there is a primitive level of skeletal system. Now, don't compare this with the organ system. Okay, it has a cellular level of organization only. But I am telling just a kind of example. It has a type of skeleton made up of spicules and spongin fibers. Sexes are not separate, that is, they are hermaphrodite. Eggs and sperms are produced in the same individual. This is very important point. Sponges are hermaphrodite. Here, eggs and sperms are produced in the same individual. Sponges can reproduce asexually by fragmentation and sexually by formation of gametes. And fertilization is internal. Fertilization is internal. Here is the development is indirect, that is through a larval stage. Development is indirect, that is through a larval stage, which is morphologically distinct from the adult. Which is morphologically distinct from the adult. Now we are coming to phylum Coelenterata or Nidaria. Now we are coming to phylum Coelenterata or Nidaria. So this is the last phylum of today's class. After this, we will continue with this kingdom Animalia in the next class. They are aquatic, mostly marine. They can be sessile as well as free swimming, and they are radially symmetrical. This is what I had drawn in that radially symmetrical thing. These were the tentacles. It can be divided like this, this or this into equal halves. It can be divided through all the planes into two equal halves. Let me clear this image. This consists of nidoblasts or nidocytes. These organisms consists of nidoblasts or nidocytes. Actually, basically from where this Nidaria name came, okay? It is also known as Coelenterata, and this Nidaria name came because of the presence of nidoblasts or nidocytes. See, this is the diagrammatic representation of nidoblast. And what nidoblasts have? It has nematocysts. It has nematocysts or stinging capsule, this one. They have nematocysts or stinging capsules. And as I told, hence the name Nidaria. What are the functions of nidoblasts? It is used as defense because we can see it has stinging capsule. So it is the defense or method of Nidaria. It is used for anchorage. It can be used for capture of prey. It can be so. It is a defensive as well as offensive tool of Coelenterata, and it is also helping in anchorage of Nidaria or Coelenterata. It has a tissue level organization. Unlike Porifera, it has a tissue level organization, that is similar type of cells performing similar type of functions are grouped into tissues, and they are diploblastic. Diploblastic means absence of mesoderm and presence of undifferentiated mesoglia. So this is diploblastic. It has a central gastrovascular cavity. It has a gastrovascular cavity where there is a single opening. Mouth on hypostome. It has a single opening mouth, not like two, mouth and anus. No, it has a single opening mouth on the hypostome. Digestion is both extracellular and intracellular. Digestion can be of both types. Here, these corals have skeletons composed of calcium carbonate. The corals have skeletons composed of calcium carbonate. Last time in Porifera, of what the skeletons were formed? It were formed of sponges. It were formed of sponges. And here it is formed of calcium carbonate. Two basic body forms are present: polyp and medusa. Polyp and medusa. Polyp is what? Two types of body forms are there. First one is polyp. This is the polyp form. This is the polyp form. This is a tube-like structure. What are its characteristics? It is sessile, cylindrical. Like I told, tube-like structure, it is sessile. Sessile means it is not free swimming. Sessile means it is not free swimming. It is sessile and cylindrical. For example, Hydra and Adamsia. This is the picture of Adamsia. This is the picture of Adamsia. And Medusa. Medusa is umbrella shaped and free swimming. Medusa is, see here, this one, umbrella shaped, and it is free swimming like Aurelia or jellyfish. Like Aurelia or jellyfish. So there are two body forms. Now is the fun. What is metagenesis? Metagenesis is present, like that phenomena in which a single cnidarian has both polyp and medusa phase in its body forms. Understood? It has both polyp and medusa in the same organism. So this phenomena is known as metagenesis, and which is a type of alternation of generation. Now how it is formed? Polyps produce medusa asexually. Like for example, first the body form is polyp. Okay, it has a cylindrical body form. It produces medusa from it asexually, maybe by fragmentation. Somehow it is by budding, it is producing medusa asexually, and medusa produces polyps sexually. Medusa produces polyps sexually. So example is Obelia. Example of this alternation of generation containing Nidaria is Obelia. Okay, these are the few other examples of Nidaria or Coelenterata. So today we are ending our session here, and again I'm telling you, you can comment on my videos, like telling me which are the chapters which are more important for you or you want me to teach. Anyways, after this I'm going to teach directly structural organization in animals. Okay, I think that would be a very important chapter for from the NEET perspective. So, and if you like my videos and if you want to encourage me to make more videos, then you can like, share, subscribe my channel. And thank you, students. Till the next video, happy learning. All the best. Bye-bye.